I hope my screen is visible to all and also I am audible to all.
Yes.
Yeah.
Shraddha.
So I'll start my session with
1 minute. 1 minute.
introduction. Okay.
1 minute. 1 minute.
Yeah. Yes, sir. No problem.
Yes. At the outset, I'm very happy today to start one more lecture in PhD webinar series. The main objective of our series is to give a platform to the PhD students of India so that what research they perform, let other inspiring students should know and they can follow. This lecture is recorded on YouTube, available forever, and definitely it will fill the gap in between the teachers who have done the research and teachers who are aspiring for the research. So today with us is a Shraddha Kulkarni from Sinhagad College, Biotechnology Department at Pune, Maharashtra. She will present her lecture, then there will be discussions. Dr. Nilima ma'am will conduct the question. He will organize the question and answer session, and she will express vote of thanks. So I will request now, I request Shraddha Kulkarni. Please, you introduce yourself to the mob, to the audience, so that they will know what is your academic career. Thank you. Shraddha, I think Shraddha has left because of some problem. She will join. She will join. Some problem is there. In the meantime, Vaishnavi Jagtap is here. Sahil Kunkar is here. Prafulla Randive is there. Very nice. Prafulla Randive is from Mumbai, and he is interested to keep communication with Shraddha Kulkarni for her research. So very nice, Shraddha. Are you there now?
Yes, sir. Yes, sir. Yes, sir. Sorry, sir.
Close.
Sorry, sir. There was some problem with my internet, but now I'm there.
Okay.
Yeah.
Now you can present and you can introduce yourself, please.
Yes, sir. Yes, sir. Okay. So, hello all. I'm Dr. Shraddha Kulkarni, working as an assistant professor in Sinhagad College of Engineering, Department of Biotechnology since last 15 years. And I have done my BTech and MTech from KIT, Kolhapur Institute of Technology, Maharashtra. And I have done my PhD from Department of Technology, Savitribai Phule, Pune University. And topic of my research was on the production and optimization of hydrophobins from fungi. Out of that, I will be presenting a part of my thesis, which included isolation, identification, and characterization of hydrophobins from fungi, which was the major part of my research. Few things I would like to mention about this topic is hydrophobins are let us go to the presentation outline so that I can give some gist of the problems which I faced during the isolation of hydrophobins. So I selected this topic to present in front of all. So presentation outline is introduction, literature review, methodology, results, major outcomes and significance, list of publications and references. So what are hydrophobins? Hydrophobins are small amphiphilic proteins and having 8 cysteine residues specifically positioned in the structure. And they are self-assembling surface active proteins and ubiquitous to fungi. They play different roles in fungal life cycle and broadly divided into class 1 and class 2. So class 1 hydrophobins are found both in ascomycetes and basidiomycetes, but class 2 were found only in ascomycetes. If you look at Uniprot database, in last 2 decades, there is a vast research done on identification of different hydrophobins. What are the typical characteristics of hydrophobins are their amino acid sequence are not homologous, or they don't have a similarity in the sequences. So if one fungal strain is having 20 hydrophobins, each and every hydrophobin will have different amino acid sequence except this 8 cysteine residues which are placed at regular interval for class 1 and class 2. And so there is a problem of having standard protein for purification of hydrophobin, comparing its purity and its and also for finding the doing the identification of the protein. This was so in my research, this was the major part, to isolate the protein, to study its characteristics and to identify it, because standards are not available. Then most studied hydrophobin is the class 2 hydrophobin HFB1 from Trichoderma raceyi. What are the roles of hydrophobins? So basically, in roles of hydrophobins, hydrophobins are playing very beautiful roles in life cycle of fungi. If you look, fungus are widely available in the environment. They can adjust with every situation of the environment, starting from braid mold to aspergillus, different species of aspergillus. Basically, what hydrophobins are doing, they are helping fungus to bridge the water layer by lowering down the surface tension of water layer and help the hyphae to grow into the air. Also, they line the spores so that spores, wherever spores are falling, they can survive there and grow. They helping fungus to attach to different hydrophobic surfaces, as fungus can grow with very little amount of water available to them. Then also they are helping to invade the insects wherever required, and also associate with the plants for different endophytic activities. Looking at the structure of the hydrophobin, they are amphipathic in nature, means they are having hydrophobic and hydrophilic amino acid sequences in them. And these amino acid sequences, they can beautifully arrange on the surface and can make hydrophilic surface into hydrophobic and hydrophobic surface into hydrophilic. So in a way, they can be used on different industrial applications. What are the applications? They can be used in biosensors for attachment of enzymes onto the sensor. For purification of recombinant protein, they can work as a tag protein, where you can use liquid-liquid separation methods and aqueous two-phase separation methods, where specifically you can separate this hydrophobic or amphipathic protein along with your recombinant protein in the hydrophobic phase, and then further purification can be carried out. They can be used in different food forms like aerated milkshakes, then coffee, cold coffees, then in ice creams. Nowadays, companies like Unilever, they are using it in one of the ice cream brand. And also they can be used by designing drug delivery system by making the nanosuspensions specifically for hydrophobic drugs. Hydrophobic drug and hydrophobin can be combined to form, can be interacted physically to form the nanosuspension and can be delivered. So looking at different applications of hydrophobins, what are the attempts made to improve the yields? These are few major attempts, highlighted attempts made, which I found in the literature. So HFB1, as I told, it is the popular class 2 hydrophobin. Class 2 and class 1 hydrophobins, basically class 1 hydrophobins form strong rodlet structure. They are totally insoluble in detergent like SDS. Class 2 hydrophobins are soluble in SDS. Class 1 hydrophobins can only be dissolved in strong acids like 99% pure trifluoroacetic acid or formic acid or performic acid. And class 1 hydrophobins are more inclined towards attaching to the surface and changing the nature of the surface. But class 2 hydrophobins are more inclined towards foaming and stabilizing the foam. So accordingly, there are applications. If we look at HFB1, it has been used in various applications. As I told previously, for food foams, HFB1 is majorly used right now. So Trichoderma resi was the original fungal source for HFB1. Overproducing strain of HFB1 was designed, and maximum yield was 600 mg per liter. Then from Aspergillus fumigatus, Pleurotus austratus, Nudulans, Schizophilum commune, Trichoderma resi, another hydrophobin HFB2 have also been produced using different methods. And when we searched for all these organisms, we decided to go for grass organism strain, that is generally regarded as safe strain, so that we can directly purify the protein and can use it in different food and medicine related applications. So this is after searching for the literature, we found Pleurotus austratus is actually producing 40 different types of hydrophobins, but only one is studied well. So we decided to go for this strain. And Pleurotus austratus is nothing but a white rot edible mushroom. It is also called as oyster mushroom, which we regularly consume in hotels. Mushroom soup, mushroom vegetables, and the cooked vegetable we consume is oyster mushroom only. That is Pleurotus austratus. And in recent study, 2021 study, researchers have found that there are 40 hydrophobins present in Pleurotus austratus, and expression of each and every hydrophobin is happening at different life stages of the mushroom. For example, VMH, but if we look at each and every stage, VMH3 is getting expressed at almost each and every phase of the life cycle of fungi, of this Pleurotus austratus. After doing extensive literature review, we published our work in Process Biochemistry as a review, production of hydrophobins from fungi. And once we decided the organism, we went on isolating, purifying, and identifying the hydrophobin. So what were our methods? Our methods include fermentative static growth. Initially, only we carried out shaking method and static culture method. And we found that when we have grown organism in static growth conditions, we have got more growth of the organism, and production of hydrophobin was also more. So total time period required for growth of fungus was 10 to 13 days. And after 10 to 13 days growth, we separated the mycelia, and fermentation broth was collected for some preliminary studies. Preliminary studies included study of biosurfactant properties of fermentation broth, in which we have studied its foam stability, emulsification assay, oil displacement assay, which are very common methods used to screen the biosurfactant producing fermentation broths. And then once we have got some positive results for the preliminary studies of biosurfactant, presence of biosurfactant, we went on isolating the hydrophobins using reported methods. If we look for reported methods for isolation of hydrophobins, they are very unique methods apart from other methods used for other proteins, because the most important and unique characteristic of the protein is it is totally SDS insoluble. And as we have selected basidiomycete, we were expecting only class 1 hydrophobin, not class 2. Then because class 2 is soluble in SDS. So now we have isolated HPs from fermentation broth. We carried out SDS page, as it is a typical small molecular weight protein ranging from 7 kilodalton to max 20-25 kilodalton. We also have got some lower kilodalton bands. And then we carried out identification, partial characterization by reverse phase HPLC, and complete identification by Orbitrap HR LC-MSMS. Simultaneously, we have studied emulsification property of that purified protein too. So now I will discuss in detail the methods used for purification. The method used for purification from fermentation broth was depending upon forming of the foam. So this foaming we have carried out using blender, varying blender for 5-7 minutes. Firstly, when we have studied stability of foam, we knew that foam remains stable for 2 to 4 hours. So by collecting this foam, we followed centrifugation, and some aggregated proteins were pelleted down after centrifugation. These aggregated proteins were nothing but hydrophobins. We lyophilized them and treated them with trifluoroacetic acid, as I mentioned earlier. Then we sonicated these samples, because see, whenever the protein hydrophobin is in aggregated form, this aggregate is nothing but a tough amyloid or the rodlet-like structure. Okay, so we have treated it with TFA and sonication to separate the monomers from that rodlet structure. And then we have air dried the samples and dissolved in suitable solvent. Generally, we use 60% ethanol or distilled water, as it is mentioned in the literature. When this is for fermentation broth, but also hydrophobins are present on the cell walls of fungi. So we followed separation of hydrophobins from that cell wall also. So for that, we have treated the cell walls with 2% SDS, the mycelia. We treated twice with 2% SDS. This is very common conventional method. We have modified it with reference to Longobardi method given in 2012 for VMH2, where we have treated the mycelia for 3 times with 2% SDS to remove all the proteins and cellular contents present in the mycelia. The proteins which are SDS soluble, they were definitely removed when we have treated it thrice, and all the cellular content also. Then we treated it with 99% pure TFA, now to ensure dissolving of hydrophobins into it and also producing its monomer. We air dried it, and then we treated it 3 times with chloroform methanol 2:1 ratio to separate lipids which are present along with our proteins, because the protein is present in the cell wall. Then we have diluted it and adjusted its pH to 7, and we have given a heat treatment. Reading in the literature that when you give heat treatment for 10 minutes up to 80 degrees Celsius, hydrophobins tend to aggregate. So we have initiated the aggregation and carried out the vortexing. So whatever protein has been aggregated got settled down, and the proteins which do not tend to aggregate at that temperature were remained suspended. So that pellet which we have got was of pure hydrophobins. We ensured it and identified it in methods which I will be explaining subsequently. And then we dissolved it in 60% ethanol or distilled water as per our requirements. So these were my methods. Now I will discuss about the results. So this is the foam which we have produced in fermentation broth, and we have studied the half decay time profile of the foam. So what is half decay time profile? It is the time required to decrease the height of the foam to the half compared to that of the initial height. And that half decay time for the SDS, we always use SDS as a positive control because it is the well-known surfactant detergent. 0.1% SDS half decay time profile was 80 minutes, and for our foam, which is produced in culture broth, the half decay time profile was nearing to 70 minutes. Then when we have purified it using the method mentioned for fermentation broth and for mycelia, we have carried out SDS page, and considering the concentration could be low, we have stained it using a silver staining method, and we found 2 bands at 11 and 17 kilodalton, which were definitely low molecular weight proteins, and almost our gel was clear, and only 2 bands were present, suggesting the partial or near about complete purification of the protein. Then we, as I discussed, we have tried emulsification of the fermentation broth using olive oil, sunflower oil, and hexane. Hexane we have selected because it is the most hydrophobic solvent. And also we have carried out at 0 hour, and we have checked the emulsified layer at 24 hour, and we found the emulsification indices. We found the emulsification index at sunflower oil was highest, nearing about 80.9%. And for hexane, it was lower. And oil displacement assay was also performed. Total protein content was also found out using Bradford method. So after studying fermentation broth for different properties And also we have carried out the purification of HPLPs using different steps. So here I am showing you the pellets of hydrophobin formed at different stages. After completing the purification process, when we have modified it, we got good concentration of hydrophobin, and we shifted from silver staining to regular blue staining, and where we have used the G250 strain for staining, Komasi G250. So here again we have got 11 and 17 kilodalton band, and we observed that we have repeated our experiments to check the presence of these two bands only in our samples. Then we have carried out the reverse phase HPLC using C4 column specifically for proteins. And we got 3 peaks. First was dedicated for the solvent. Second, we neglected, but the third one was important for us, which we got at RT 35 minutes. Now, this was typically in between. See, this was a gradient created for acetonitrile from 10% to 90%. And at 30 to 40% gradient, we have got this peak. So this peak was important because in reported studies, hydrophobin separation on HPLC was always happened in between 30 to 50% of acetonitrile gradient. So it is the amphiphilicity or the non-polar nature. When it is getting created inside the column, there is a separation of the hydrophobin observed. Accordingly, we have also got the peak, and this RT which we have got was matching with the VMH2 hydrophobin RT. VMH2 was the hydrophobin purified from Pleurotus austratus only. So till this phase, we decided to call our protein as hydrophobin-like proteins because we did not have any confirmation about the sequence or exact nature of the hydrophobin, but it was matching all the properties of reported hydrophobins. Then we have carried out emulsification of purified protein also. As we compared for fermentation broth, we found that for sunflower oil, it has given 85% of the emulsification index. Stability was good for sunflower oil as it was for the fermentation broth. But when we have checked it for N-hexane, 42% of emulsification index was recorded. And if you compare that with SDS and water, which we used as positive and negative control, that stability of emulsion was very good when it was done with purified protein. Then we went on doing Orbitrap HR LCMSMS. My method to carry out Orbitrap HR LCMSMS was, we have separated our proteins on SDS page, we cut the band on SDS page, and we have treated that with trypsin, proteomic trypsin. After trypsinization, the small peptides were submitted to UHPLC system where they were separated and analyzed by MS. And this is the chromatogram which we received after MS. And this is for 11 kilodalton protein, and this is for 17 kilodalton protein. This is called total ion chromatogram. And at different time period, we have got some peaks. These peaks indicates the presence of the peptides there. This was the result of MS. After MS, these peaks were picked up and sent for the further MS, which we overall called Orbitrap HR LCMS MS. In next MS, these peptides were broken down into single amino acid structures, and these amino acid structures were compared, matched with the mascot software, and the sequences were derived. So these were the annotated peptide sequences. Thermo Discoverer 2.2 software was used, and under which we have carried out the XCOR HT search engine was used to compare the sequence with the reported sequences of hydrophobins of Pleurotus austratus, and only the peptide sequences were selected to find out the hydrophobin, which has shown XCOR score above 2. XCOR score above 2 suggests the match is not random, and it is the perfect confirmed match with that hydrophobin protein sequence. By doing this, it was found that our hydrophobin-like protein was nothing but VMH31, and its molecular weight, The XCOR HT search engine give us the molecular weight, PI, and all other properties of the protein also, which will help us again to confirm the same protein. And our protein was VMH31 with its molecular weight 11.1 kilodalton. And in total, we have found 12 peptides of VMH31 in a band of 11 kilodalton, which we have cut from the SDS page gel. Now, when we went on searching for 17 kilodalton protein using the same software, we found that again, surprisingly, the VMH31 hydrophobin was only present in 17 kilodalton protein, but its molecular weight confirmed was 11.1, and three annotated peptide sequences were confirmed. In total, 66% amino acid sequence. This is the amino acid sequence which I have got from Uniprot. It contains in total 108 amino acids and its isoelectric point is 7.48. So the yellow mark sequence is nothing but the signal peptide, and all these other amino acid sequences, out of that 66% sequence we could match. We have recovered that many peptides, and I'm happy to share with you all. This is first time, because for VMH2 people have only found 2 major peptide sequences for match. And in our work, we have found 66% amino acid sequence, which confirm the presence of VMH31 hydrophobin in our protein. Then why the same protein in 17 kilodalton protein sample? We have done different experiments and we searched for the literature also, and we found that hydrophobin tend to aggregate in SDS page also during the procedure, and they form different conformation and show different molecular weight, and so give the band at different molecular weight also. So accordingly, our 17 kilodalton protein has also shown VMH31. Looking for the example, ABH1 hydrophobin from Agaricus bisporus. It is also basidiomycete mushroom like Pleurotus ostreatus. Its molecular weight is 8 kilodalton, but whenever it is run into SDS page gel, it shows 8, 16, and 32 kilodalton band. Researchers have separated the band and tested for the presence of hydrophobin, and like us, they found ABH1 is only present in all the 3 bands. So it is the unusual conformation formation of hydrophobin when it is running in a SDS page. We also observed that, I can go back and show it to you. We also observed that whenever we are running the samples in gel, 11 kilodalton band was darker, and always 17 kilodalton band was faint in a nature. And if the concentration of the protein is less sometime, then it was only showing 11 kilodalton band. And we have compared it with other hydrophobins, popular hydrophobin SC3, which is separated from Schizophilum commune. It was the first hydrophobin purified. It is also showing these different conformations and different molecular weights. So we concluded that 17 kilodalton protein is no different hydrophobin. It is the different conformation of same hydrophobin VMH31. Why VMH31? Because it is an allelic protein. VMH3 has 2 alleles, VMH31 and 2, out of which we discovered VMH31. And I'm happy to share with you all, this is the first time reporting of the hydrophobin VMH31 at protein level. Genomic level, it is already mentioned, and its genomic sequence is also provided. Some roles are also predicted, but nobody has purified it from Pleurotus ostreatus. We are the first to purify it from Pleurotus ostreatus, also to sequence it properly. And in further studies, we have first time characterized the protein also. These are my OHR LC-MS spectra of tryptic peptides. This is the summary of table of purification process. So if we look at the purity, 80.85% of purity we have got, and recovery was less. Actually, now in my future studies, I am focusing more on increasing this recovery by using different purification methods and trying to avoid use of TFA, trifluoroacetic acid, so that it can be more easily incorporated in biomedical applications also. So once we knew that we have isolated VMH31, which is the first time purified hydrophobin, it was our responsibility to characterize the hydrophobin, and we were curious to know how hydrophobin is playing role in changing the structural properties, and also does it have any functional property. So for that we have designed our methods like this. Secondary structure analysis was carried out to know what are the structural characteristics of the protein by using CD spectroscopy tool, and we have used Capito tool to analyze that uh, Excel readings which we have got. We have submitted that to the Capito tool, and we have got a graph which is interpreted by the Capito tool to know how much alpha helix, beta sheets, and random coils are present in the protein. Then theoretical structural analysis was also done using FIRE2 bioinformatics tool. Hydrophobic cluster analysis was carried out using HCA analysis tool, and glycosylated nature was also studied. When the hydrophobin VMH3 was previously studied at genetic level, genomic level, people have predicted that it must be glycosylated like SC3. SC3 is the hydrophobin from Schizophilum commune. These 1, 2, 3 nomenclature given to the hydrophobin depend upon its role. So when it is SC3 and VMH3, that 3 suggests they have got something similar in their role in their own species. That is, whatever SC3 is playing a role in Schizophilum commune, VMH3 must be playing a role in Pleurotus osculetus. So if SC3 is glycosylated, what about VMH31? We have found out that also. Then we have studied its surface active properties by finding out water contact angle, determination on hydrophilic surface and hydrophobic surface, and interaction with hydrophobic surface. So analysis of VMH31 was carried out, analysis of behavior in solution. So what happens if we provide different percents of ethanol solutions? 1 second, 1 second. So different percent solutions of ethanol were used, 20, 40, 60, and 80, to check in which our protein is stable. See, because previously when we are treating it as hydrophobin-like protein and using 60% ethanol and water for the process, we are just using both the solvents by studying it in a literature. But now we were curious to know what is the stability of the protein and which solution is good for the protein. Also, we have studied what is the effect of different pH, because for VMH2 and some other hydrophobins, as I already mentioned, different temperatures and different pH values have shown effect in aggregation of the protein. So we have carried out that experiment also. And different bioinformatics approach like physicochemical properties we have studied using XPASI protparam. Phylogenetic analysis was studied using molecular evolutionary genetic analysis tool, that is MEGA tool. And hydropathy pattern was also studied using XPASI ProtScale tool. So this was the protocol for water contact angle determination on glass and Teflon. Why we selected glass and why we selected Teflon? Teflon as a hydrophobic surface, glass as a hydrophilic surface. Okay, and protocols are mentioned here. Protocols are slightly different for Teflon and for glass, but when we have treated Teflon, at one step we have treated it 2% SDS. So we have cleaned the Teflon surface, then we have coated it with VMH31 at the concentration of 50 microgram per ml, incubated for 16 hours, and then once we have coated it with VMH31, we have treated it, boiled it in 2% SDS, and then we have dried it again and used water drop to be placed at 6 positions, and we have captured the images. We used ImageJ software and LBSDSA plugin to check the water contact angle, and mean water contact angle of 6 drops was recorded. So when we have treated it with 2% SDS and still when it has given us the results, indirectly we have confirmed that our protein is class 1 protein, class 1 nature protein. For glass, the treatment was more or less same, but few steps were added as the excess water content was removed by incubating in 60 degrees Celsius hot air oven, etc. So let us go to the results. In my results, we have got, this is a CD curve of my protein, hydrophobin VMH31. I have submitted it to CD spectrometry analysis. And this is the output given by Capito tool. This is non-smoothed curve I'm showing here. And the interpretation of the CD curve by Capito tool has shown that in total 2% alpha helix, 51% beta sheets, and 47% random coils were present in my protein. Now, Capito tool is also giving us double wavelength pattern or double wavelength comparison, where they compare the wavelength at 200 and 222 nanometers to check where the protein is present as a structural, structural variant. So if we check, protein could have native structure or the molten globule structure, pre-molten globule structure, or unfolded structure. We all learn in biochemistry that there is a native structure formation, primary structure, then secondary, tertiary, and then quaternary structure. But in actual functioning of the proteins, different proteins are present at different stages when they are functional. So it is not necessary that we should have our protein is in a native format to fully function, to remain fully functional. About hydrophobin, they are called as IDPs, intrinsically disordered proteins. So whenever they are functional, they are not perfectly in the native form. They are in pre-molten globule form. Means they will have more random coils, more beta sheets, and very less alpha helix. Okay. So when they are in native structure, maybe alpha helix is more, beta sheets is balancing that, and random coils could be very less. But this typical structure of hydrophobin, when we have got after our experiments, we compared this with literature and we found almost all hydrophobins, SC3, ABH3, HFB4, HFB7, each and every hydrophobin falls in pre-molten globule, this red dot shown here, pre-molten globule region. Means these proteins are not perfectly folded in a native form, and then only they function properly. So when we compared it and we found our structure of our protein is also falling in that category, one more confirmation we got. We theoretically compared the structure using the XPASI protparam, as I told before. It is a bioinformatics tool. It only predicts the structure by getting the protein sequence. I have provided FASTA format of amino acid sequence, and here we found alpha helix 14%, beta sheets 37%, but still disordered form is 48%. Then why this difference? So generally, when hydrophobin is binding to any surface, its alpha helix nature is increasing. And when it is in a solution, its alpha helix component or the alpha helix contributing part in the structure is less. So we have provided it in a solution for the CD spectra analysis. Our alpha helix percent was less. Then hydrophobic cluster analysis of VMH31 was also carried out to check how the hydrophobic clusters of different hydrophobic amino acids are forming and where they are present. So they are more present at the initial point, which is a signal peptide, then 50 to 70 amino acids region, and then last 90 to 100 amino acid regions. These typical structures are nothing but the hydrophobic patches. So when we have measured the contact angle, as I have already explained the procedure on Teflon surface, and when we measured it on glass surface, this is the A is the water drop on the bare Teflon and bare glass surface, and B and C are the two examples of drops which I am showing the images. I have lot many images of the drops after treating the surface with VMH31. And we found that this in this study, the VMH31 purified from culture broth has increased the water contact angle by 87% on glass, and VMH31 purified from mycelia increased the water contact angle by 70% on glass, which suggests hydrophilicity was shifted to hydrophobicity. And for Teflon, it has decreased little bit, that decrease was not much, by 17.7% and by 13.04% by the hydrophobin purified from culture broth and from mycelia. So overall, we can say that our hydrophobin was capable of changing the nature of the surface from hydrophilic to hydrophobic and hydrophobic to hydrophilic, but the result was more prominent when it was checked for glass. And the p-value also suggested that there is a statistical significance in the conversion of the nature of the surface. Then when we have carried out the analysis of VMH31 in aqueous solutions for months, it could remain stable in 60% ethanol also and distilled water also. When 20, 40, 60, 80% ethanol and distilled water were checked for the lyophilized powder of the hydrophobin we dissolved in different solutions, the concentration was predetermined, and we found that 60% ethanol was the good concentration, was the good solvent for dissolving the hydrophobin. When turbidometry analysis was carried out, we found that at pH 4 there was a little aggregation of the protein, but from pH 6 there was a start of aggregation. Until pH 9 there was a maximum aggregation of hydrophobin. When we say aggregation of the hydrophobin, in case of VMH2, people have studied that there is a formation of more beta sheets. So formation of beta sheets and layering of beta sheets on each other helps for aggregating of hydrophobins due to the change in pH. To study the glycosylated nature, our observation was whenever we were dissolving our protein into 60% ethanol, we were getting undissolved, we were getting a pellet. And once we take out the solution for SDS page analysis and anything for the hydrophobin, that pellet we tried to dissolve in water and it was not dissolvable. So we directly treated it with the phenol sulfuric acid test we carried out, and we directly added sulfuric acid and phenol. We added phenol sulfuric acid reagents to check the presence of the carbohydrates, and we found that there was presence of carbohydrates in the pellet, suggesting glycosylated nature of VMH31. And whenever VMH31 was dissolved in 60% ethanol, this glycosylating carbohydrate molecules were separating, dissociating from the protein and remaining as a pellet in the sample. Then we have studied physicochemical characteristics using EXPASI. And these were the characteristics. Molecular weight we have already confirmed in OHR, LCM, SMS, and in our SDS page analysis. Theoretical PI 7.48. Then aliphatic index, instability index, and GRAVI. GRAVI is nothing but grand average of hydrophobicity. So hydrophobicity of VMH31 was comparable with some other hydrophobins, but not that much. It is just more than that of HYD1. If you compare it with VMH1, BH3, VMH22, and SC3, these are more hydrophobic than VMH31. Instability index, the EXPASI tool mentioned that, or they define as, if instability index is below 40, that protein is more thermally stable, and that we have already experienced because in our protocol we have added 80 degrees Celsius step and protein is absolutely fine. Aliphatic index dependent upon the what type of aliphatic amino acids are present, and that again give the stability to the protein. So finally, we have carried out phylogenetic analysis of the protein to check the evolutionary placement of the protein and its classification. And also we have compared the hydropathy pattern of VMH31. In phylogenetic relationships, I am showing you maximum likelihood method graph, which I have collected from Mega tool. And VMH31 is closely placed with VMH1, VMH2, and ABH3. VMH1, VMH2 are from same species, and ABH3 is from Agaricus bisporus mushroom. Now, I would like to clear here that this relationship is more based upon roles of the hydrophobin and how they are closely related with each other, and not exactly with the sequence. When you compare with the sequence, the SC3 and VMH31 are, if we look at, they are far from each other, but they play similar role. And VMH1, VMH2 are close to VMH31. We have added some class 2 hydrophobins in our study, phylogenetic relationship study, to compare the class 1 nature of my protein. And if we can observe this club of class 2 hydrophobins are far away from VMH31, and all these are the class 1 hydrophobins. So again, it was a confirmation that my protein is class 1 hydrophobin. Hydropathy pattern study was also the same thing, where we actually give a score to the amino acid. Hydrophobic amino acid will have positive score, hydrophilic amino acid will have negative score, and the pattern of hydrophobicity is drawn here. So hydropathy score on the y-axis and the number of amino acids on x-axis, and we can see that the pattern was very much similar and comparable of VMH31 with all the other hydrophobins, VMH1, HYDPT, SC3, etc. So this is my overall summary of the study. We have studied VMH31 using different computational techniques and also using experimental techniques. In my computational techniques, I found that there is a 37% beta sheets, 14% alpha helix, and 48% random coils. But in actual experimental, laboratory experimental study, it was 51% beta sheets, 2% alpha helix, and 47% random coils. Phylogenetic studies and hydropathy pattern confirmed the class 1 nature. And in our experimental studies, the SDS insolubility verified by treating Teflon surface with 1% SDS coated with VMH31, and no change in WCA confirmed the SDS insoluble nature, that is class 1 nature. Gravity index and hydropathy index suggested the amphipathic nature when it was done using computational studies. When it was done using experimental studies, VMH31 altered WCA on hydrophilic and hydrophobic surfaces, exhibiting the amphipathic nature. So these are my major outcomes and significance of this part of the study. Hydrophobin VMH31 was first time isolated and purified from white rot mushroom, Pleurotus austratus, grown in static liquid culture conditions. Then concentration levels of VMH31 were higher when purified from mycelia compared to secreted protein isolated from culture broth, suggested less secretion of VMH31 in the culture medium. Molecular weight of VMH31 was found to be near to 11 kilodalton, confirmed by performing PAGE and OHR LCM SMS analysis done using the Sequus HT search engine. Protein has shown typical emulsifier activity by stabilizing emulsions formed with food grade oils and amphipathic nature by altering surface properties of Teflon and glass. Protein has shown stable concentration in 60% ethanol and double distilled water for more than 20 days. Turbidometry analysis performed for the effect of pH helped to understand that alkaline pH encouraged aggregation of VMH31 in a solution. Evaluation of hydropathy plots and evolutionary studies helped to confirm the placement of VMH31 in the hydrophobin class 1 family. After studying the protein extensively, we have presented our work in Protein Expression and Purification Journal in August 2022. And title of our paper was Identification and Characterization of Hydrophobin VMH3 from Pleurotus austratus. These are my other publications from my PhD. These are my conference presentations up till now. Few references. Thank you. I rest my presentation.
Thank you, ma'am. Thank you for the great session. We'll be extending vote of thanks after question and answer session.
Yes, ma'am.
I do here with honor Dr. Arvind Deshmukh sir for chairing the session. Dr. Prakash Alami sir, the esteemed scientist from CFTRI. He has asked the few queries like, ma'am, can I ask?
Yes, ma'am. Yes, ma'am.
Yeah. What is asking is how you scale up production and the commercial availability and utilization.
Actually, for scale up, we have tried few methods. We have basically designed media for production, because our observation is in the literature, defined media is not present for hydrophobin production. People are more focusing on isolating new molecules and studying its properties. So we have designed a media, and we have also studied most affecting parameters on the process by designing mathematical model. Unstructured kinetic model we have designed, and we have found out most important parameters affecting the process. And right now we are in a process of upscaling till 10 liters, and we have optimized the process and enhanced the yield by 6 times by doing all these efforts.
Okay, great, ma'am. Thank you. Yes,
Yes,
you
ma'am.
have design media. You
Yes.
are going for mathematical models and are going to upscale it 6 times more.
Yes.
So next question asked by him, ma'am. What is the novelty of hydrophobin that has been reported in this study?
Novelty of hydrophobin is basically, as I mentioned in my presentation, this is first time isolated at protein level, this hydrophobin, VMH31. And so whatever properties we have studied is novelty of that protein. So as I have mentioned, for glass, its property was more enhancing the surface change, means it is more active in changing the hydrophilic surface from hydrophilic to hydrophobic, and not comfortable with the hydrophobic surface by converting hydrophobic surface into hydrophilic. So if you look into the literature, these properties of each and every hydrophobin are different, ma'am. So VMH3 has shown some specific activities which I have mentioned in my talk.
Yes, ma'am. Thank you for enlightening us with respect to this question. To the audience, anyone would like to ask question with reference to the topic? You can directly ask to ma'am also,
Yes, yes.
or you can chat.
If there is no any question,
Sir,
Nilima
one
ma'am.
question I can see here.
Okay, okay.
Just I'll go to the chat. Can we isolate hydrophobin some bacterial? Yes, I just can't tell you exactly the name of bacteria which is used to isolate hydrophobin. Not exactly hydrophobin, because hydrophobin is specific to fungi, but hydrophobin like proteins have been isolated from bacteria. Right now, I don't know exactly the name of the species, but we can. Surfactant is the hydrophobin like protein isolated from bacteria. We can. Hydrophobins are also involved in interaction phenomenon. Yes, interaction phenomenon means fungal interaction with plants and insects. Like if you are asking, yes, they are involved. They help to attach to hydrophobic surface. So insect cuticle, when it is hydrophobic in nature, they help fungus to secrete that type of enzymes, chitinases like enzymes, which can help to invade the insect surface. Yes, they have role.
Can we go for vote of thanks, sir? Yes, ma'am. There is next question. How
Yes,
much
yes.
is proteins in amino acid length?
Actually, my protein is 108 amino acid in length. And do you think they are in the form of genes? Yes, different genes of hydrophobins are present, and due to slight changes in the genetic sequence, they are in the allelic form. As for VMH3, it is VMH31 and VMH32. Maximum 100 to 150 amino acid sequences are present in different hydrophobins. Hydrophobins they are not only involved in structural components, they have functional roles, as I explained, to bridge the water surface by lowering down the surface tension. Layering the spores means they play some structural role which has got some function. That is the peculiarity of the protein. Okay, so if fungus is secreting hydrophobins to lower down the surface tension, this is the function of the hydrophobin. But when they layer the spores, layering is the structural part. It is getting layered onto the spores, but it is playing the functional role to help the spores to survive anywhere, making the spores tough. It is like that. Yes, they have introns too. Yes. And that placing of the introns and splicing, etc., decides the different types of hydrophobins and their evolutionary developments. Yes.
There is one more question.
Hydrophobins are not used as biosensors. They are used to fix the enzyme onto biosensor surfaces to give more stability to enzymes, attaching of the enzymes. I hope I have cleared all the doubts.
Yes, so can we go for vote of thanks?
Yes,
Deshmukh
yes,
sir?
yes.
Yeah. For this weekly PhD webinar series on every Sunday, we do have, and it was 27th session. And the 27th session, it was resourced by Dr. Shraddha Kulkarni, who is from Department of Biotechnology, Sinhagad College of Engineering, Pune. And her topic of research was identification and characterization of hydrophobin from fungi. Hydrophobin, which is an emulsifier so as to stabilize emulsions and which is amphipathic in nature, was explained by ma'am very nicely. And for this session, we had in the chair as chairperson Dr. Arvind Deshmukh sir, who is President Microbiology Society India for India, as well as there is elite presence of Dr. Prakash Halami sir.
Hello, ma'am.
I think some technical problem is there.
Some technical issue.
Hello. Okay, then let us depart today with extensive knowledge on hydrophobin. And
Thank you.
definitely it is recorded. It will be made available from tomorrow on YouTube.
Okay, sir.
And Shraddha ma'am will receive
Hello.
one memento from
Okay.
Microbiology Society.
Thank you,
Am I
sir.
audible? Am I audible, sir?
Yes, ma'am. Yes, ma'am.
Yes.
Yeah, yeah. It was network failed.
Hmm.
So, ma'am, she has enlightened the ma'am. She enlightened us with the fermentation broth, that is static method she followed with. She went for cell wall of fungi, hydrophobin recovery. She looked into by using the different oils or hexane like olive oil and sunflower oil, and found out for the stability of emulsion using Orbitrap HR LCMS. She detailed, she worked out with the proteins, peptide sequences, amino acid sequences. She annotated and molecular weight she found of VMH31. It was 11 kD. And she told us that in case of Floratus astratus, it was hydrophobin, this VMH31. It was found in 17 kD Dalton band also, and showed for the first time that it is, yes, allelic protein. And it was really wonderful, ma'am,
Thank you,
to
ma'am.
get through this particular knowledge. And in future, you are going to improve purification as per
Yes.
query which has been answered. And you went through wonderful studies, ma'am, for the characterization of
Thank
hydrophobin,
you.
the structural characterization with respect to bioinformatics, also functional characterization you went through. And
Yes, ma'am.
Very good.
Yes, sir.
Yeah.
hello,
Hello, sir.
you told us that it is amphipathic nature of this protein, and you went for computational studies, phylogenetic analysis, gravy, as well as water contact analysis assay. You went through water contact angle analysis. You went with respect to Teflon
Yes.
and glass surfaces. Yes, ma'am, it was, yes, you concluded that it is a class 1 nature of this hydrophobia VMH31. So in
Yes,
a
ma'am.
nutshell, we could undergo through the venture of Capito tool, what are they and how they can be
Used.
explored and extrapolated into our PhD studies,
Yes, ma'am.
interpretation of CD spectroscopy analysis, and etc. Ma'am, thank you
Thank
very much
you.
once again
Thank
on
you.
behalf of Microbiology Society India. Thank you,
Thank you,
ma'am.
ma'am. Thank you.
Thank
I am also thankful to all of you. Deshmukh sir, Pendharkar ma'am, Halami sir, Krushanjali ma'am, then all the other dignitaries, because I got a platform to present my work and I could reach to the audience, students who will also get to know about the new area which they can explore and work in. So I feel very nice by getting this opportunity and very much thankful to all of you. Thank you.
Thank you.
thank you.
Thank you all.
Thank you,
Thank
sir.
you. Thank you, sir.